Hey everyone, and welcome to this explainer. Today, we're not just looking at biology and chemistry as some dusty textbook subjects. We're looking at them as one massive, continuous, highly efficient processing plant. We're going to explore how biological entities, like you, me, and the plants right outside your window, are literally walking, breathing chemical laboratories. It's absolutely fascinating when you start connecting the dots, so let's dive right in. Okay, here is our roadmap for today. 1. Human biological systems. 2. Plant tissues and photosynthesis. 3. The science of mixtures. 4. Separating with chromatography. And 5. Acids, bases, and pH. Section 1. Human biological systems. The body's processing plant. Think of the human body as an absolute marvel of mechanical and chemical engineering. It is the ultimate processing plant, right? To keep it running, we go through 3 primary stages of nutrition. First up is ingestion, taking food into the mouth where mechanical digestion kicks off with chewing and gets lubricated by your saliva. Next, we have digestion. This is the breakdown of those complex molecules into simple, soluble stuff using very specific enzymes and gastric juices. Finally, absorption. That's where those simplified nutrients pass right through the intestinal wall straight into your blood and lymph systems for distribution.
Now, inside this processing plant, chemical digestion isn't just throwing things into a random mixing pot. No way. It has incredible specificity. In your mouth, the enzyme salivary amylase specifically targets starch, turning it into maltose. Down in your stomach, pepsin targets proteins, yielding polypeptides. Your pancreas secretes lipase to tackle lipids, giving us fatty acids and glycerol. And in the small intestine, maltase breaks that maltose down into glucose. It's like every single enzyme is a perfectly shaped key for a very specific nutritional lock.
So, once we have fuel, we need oxygen to burn it. And this perfectly illustrates the mechanical physics of breathing. To draw air in during inspiration, your intercostal muscles contract, your ribs pull up, and your diaphragm actually flattens out. This increases the volume of your thoracic cavity, which drops the pressure inside and essentially vacuums air right into your lungs. Expiration, that's the exact reverse. Muscles relax, volume decreases, pressure spikes, and the air gets pushed right back out. Pure, elegant physics in action.
Of course, every single processing plant generates waste. That's where your urinary system comes in. Your kidneys act as the ultimate filtration system. Ureters act as the transport pipes. The bladder is your holding tank, and the urethra handles the final elimination. But wait, here's a crucial distinction. Excretion specifically refers to removing metabolic waste, things like carbon dioxide, urea, and uric acid. Feces, on the other hand, is simply undigested material. It's actually not considered metabolic waste because it never truly entered your body's internal cellular metabolism. Kind of mind-blowing, right? Section 2. Plant tissues and photosynthesis, the primary producers. So where do all those complex raw materials we eat actually come from in the first place? Plants. And just like us, plants have different cell types organized into distinct tissues. When we look closely at these structures under a microscope, from the tightly packed epidermal cells to the much more porous open structures, it's incredibly clear how biological form follows function. A tissue is just a group of cells with a shared origin modified to do a particular job. That could be providing physical structure, transporting water, or capturing sunlight. And to actually prove that leaves are actively capturing sunlight to manufacture food, we can run a classic starch test. If you are documenting this objective in a lab book, here is your procedure. First, we boil the leaf in an alcohol water bath. We have to use a water bath because alcohol is super flammable. This step dissolves the green chlorophyll. After carefully washing the leaf, we add a few drops of iodine solution. For our observation, if starch is present, the invisible becomes visible, and we get a highly distinct dark purple or blue color change. We can use that exact same starch test procedure to figure out the required ingredients for photosynthesis. If we cover a section of a leaf with black polythene and leave the rest in the sun, only the exposed part turns purple with iodine. The conclusion? Light is absolutely required. What if we put a leaf in a bag with potassium hydroxide? Well, KOH absorbs carbon dioxide from the air, and that leaf won't produce starch either. It beautifully proves that both light and carbon dioxide are absolute non-negotiables for plants to make food.
Section 3. The science of mixtures, combining the ingredients.
All right, we've seen the biology, but what about the underlying chemical rules?
In chemistry, the word mixture has a very strict definition. It's a blend of two or more pure substances where the individual components actually keep their distinct identities. For example, if you were to grind up hydrated copper sulfate and naphthalene mothballs together, it might look like one single powder to the naked eye. But chemically, they haven't bonded at all. You still have your copper sulfate and you still have your naphthalene.
And this brings us right to the concept of composition. If you dissolve 0.2 grams of potassium permanganate in 50 milliliters of water, you get a light, pale purple solution. Double that solute to 0.4 grams in the exact same volume of water, and wow, the color becomes intensely dark. Why does this matter? Because mixture composition is everything. Whether you are mixing weedicides for a farm, manufacturing pharmaceuticals, or prepping lab solutions, getting the precise amount of solute particles per unit volume right dictates the entire chemical behavior.
Section 4. Separating with chromatography.
So, if we can mix things together so easily, how do we pull them back apart? Well, paper chromatography is this brilliant laboratory technique we use to separate and identify non-volatile components. Here's how it works with a plant leaf. You crush up a spinach leaf, extract the chlorophyll, and place a tiny drop onto a strip of filter paper. Then, you suspend that paper so just the bottom edge dips into a solvent, like acetone. As the solvent evaporates, liquid slowly creeps upward along the paper, carrying the plant extract right along with it.
To understand why those components separate, we just have to look at two distinct phases. We have the stationary phase, that's the paper strip itself, and the mobile phase, the moving liquid solvent. As our mixture dissolves into the mobile phase, its various components have completely different levels of attraction to that stationary paper. Components that are highly attracted to the paper, they move upward pretty slowly. The ones less attracted race up much faster. Those different speeds cause this invisible mixture to visually break apart into distinct bands of color, literally right before your eyes.
Section 5. Acids, bases, and pH, the chemical foundations.
To fully wrap up our understanding of this laboratory of life, we need to look at the bedrock chemistry governing these reactions. Let's lay out some defining characteristics. When looking at common lab acids, think hydrochloric, nitric, or sulfuric acid, there is a glaring pattern. Hydrogen is a key component element in every single one of them. Bases, on the other hand, are a bit more varied. They can be solids or even gases, like ammonia. But in the lab setting, we usually use them as aqueous solutions, like dissolving sodium hydroxide in water.
Because we obviously can't tell what a clear liquid is just by looking at it, and you definitely shouldn't taste it, we use chemical indicators. Blue litmus paper turns red in an acid, but red litmus turns blue in a base. Phenolphthalein stays completely colorless in an acid, but drop it into a base and boom, vibrant pink. Methyl orange goes red in an acid and yellow in a base. These basic tools give us a fast visual cue about the chemical nature of whatever solution we are working with.
But to get a truly precise measurement of just how acidic or basic a solution is, we turn to the pH scale, ranging from 0 all the way to 14. Dead center at 7, you've got neutral substances, like pure water. As you drop down from 6 to 0, acidity increases dramatically. As you climb up from 8 to 14, basicity shoots up. By dipping a multi-indicator pH paper into a solution and matching it up to this color code, we immediately grasp the strength and the reactivity of the chemicals in front of us.
Which brings us to the very end of our journey. And I want to leave you with a final thought to ponder. We've seen how enzymes precisely digest your food, how your lungs exchange gases using pressure vacuums, how plants use light to physically build the food we rely on, and the underlying chemical rules that govern the whole shebang. So, knowing what you know now, how are these invisible chemical mixtures, these constantly fluctuating pH levels, and these biological processing plants interacting inside your own body at this exact second? It really makes you appreciate the incredible, nonstop laboratory of life you carry around with you every single day. Thanks so much for joining me on this explainer. Keep asking questions, and I'll see you next time.